Hello everyone. So today we are starting another video. In the previous video we studied about the cell cycle. Cell cycle, as I said, is the phase from one cell to the formation of another cell. Cell cycle completes in two important phases. That is, first is interphase and second is M phase. Interphase itself was divisible into three parts. That is the G1, S and G2 phase. And G1, in which the cell prepare itself for the formation of new set of DNA or other uh, chromatin material, and S phase in which the chromatin materials are formed, and G2 phase in which the cell is preparing all its required cell organelle and etc. for the new cell, and M phase is actually a phase of division of this. So till then we already studied. The link I will give in this I button as well as in the comment section. Now today we are studying the M phase. First one is mitosis. There are two type of M phases, the mitosis and meiosis. In this video we are going to study about mitosis. So we'll concentrate what is mitosis, what is it, where it take place, what are its steps like karyokinesis and cytokinesis, what are the phases of the karyokinesis, that is prophase, metaphase, anaphase and telophase, what are the changes taking place in each. There are a number of changes taking place because of which we are calling them as a separate name, prophase, metaphase, anaphase and telophase. And then what is cytokinesis, where it take place, how it take place in plants, how it take place in animals, what is the difference in them, and then significance of mitosis. Then if mitosis is there, then why meiosis is there? So there is some difference. What makes these differences there? What makes the mitosis so important than the meiosis, or what makes the meiosis important than the mitosis? All these we'll study in this video. So first let us study how mitosis, that is how it occurs. So mitosis is completes in two steps. The first one is the division of nucleus, and second is division of cytoplasm. We know that the cell has got protoplasm, and protoplasm is divisible into two parts, that are nucleus and cytoplasm. Nucleus, which contain the entire genetic material, and cytoplasm, which contain all the cell organelle. So when a cell divides, it should have genetic material as well as cell organelle both. So first the nucleus will divide and then this cytoplasm will divide. So this is what the two phases. Then first let us talk about the karyokinesis. The karyokinesis is the division of nucleus, and division of nucleus itself completes in four phases: the prophase, metaphase, anaphase and telophase. Okay, what are the changes taking place in this? We shall study one by one. The very first thing is prophase. So here now our main part comes. Okay, so what is happening in the prophase? Prophase, we already studied that the before cell undergoes F phase, it has to undergo interphase, and interphase is the phase in which the genetic material or the DNA content is doubled. Okay, so we know that each cell has got certain chromosomes. One chromosome looks like this. In common, we draw a chromosome like this. Okay, when the DNA content is double, when we say that the new chromatids are formed. So see, this is the chromatid which is formed here by the duplication or replication of DNA, and these two parts are now called sister chromatids. These two are called sister chromatids. So here you can see these two are sister chromatids, these two are sister chromatids, these are sister chromatids, and so on. So sister chromatids are actually formed in the S phase of interphase, and in the interphase, S phase of the interphase, the new set of DNA are formed, and these DNA are actually formed by the replication, which is doubling the DNA content or forming the sister chromatids. Now, sister chromatids are the raw form of chromosomes. As long as they are found attached to this attached at centromere, they will be called as one entity. Two sister chromatids will be called as one entity, as one chromosome. And when they will be separated, the sister chromatids separate from each other, then they will be separately known as single chromosome. So that makes a difference. Now, here the cell is undergoing prophase. So prophase means, as you know, that these were the centriole or centrosome. The two centrioles forms a centrosome. Both the centrosome or both the centriole were together here. Now they are started separating. The nuclear membrane is started dissolving. You see the nuclear membrane is shown in dotted structure. Okay, and the nucleus is already removed, and the chromatin have start becoming thick. So as the chromatin become thick, now they will be called as chromosomes. So here you see in the karyokinesis prophase, what are the changes taking place? Very first thing is nuclear membrane start dissolving. Nuclear membrane start dissolving. Second thing is the nucleus also dissolves. In a nucleus there may be one or more number of nucleus. They also dissolves. Okay. The third thing is the chromatin material start condensing by coiling. The chromatin material, as we know that the DNA is very fine and thin, which cannot be seen by normal microscope until unless we have got electron microscope. The same DNA is now coiling and coiling and coiling and becoming thick and short. As it becomes thick and short, they are now called as chromosome. The chromatin is just a fibrous structure of DNA and protein. Chromosomes are well-defined structure, okay, which will be stained and which will be able to see under the microscope. So they become short and thick. Okay, and the chromosomes now we call as chromosome. Centrosome. The centrosome divide into centrioles and they start moving away from one another, and they will form spindle fibers. Okay, chromatin material are moving towards center, or other you can even say chromosomes, and the spindle fibers are formed within the centrioles. The centrosome divided into two centrioles. The centrioles were started moving from each other, and they started forming centrioles, as I am showing this diagram here. So, sorry, we'll be able to see it in the next diagram. So here the spindle fibers, okay, between the two centrioles. So here you can see both the centrioles are reached onto the opposite poles, and the spindle fibers are formed. You can call them spindle fiber, you can call them astral fiber. Different books, different names are given, but the things are same. And we will study in the higher classes. That is, the centrioles are actually made up of microtubules and microfilaments, and these microtubules and microfilaments actually form fibers. Okay, so here, and they have an ability of contraction. So these are the all things which we'll study in the next coming chapters or next coming classes. Here we'll concentrate only up to this. So now what happens in the metaphase? During this phase, the chromatids start reaching toward the center of the cells. You see all the chromosomes are now come at the center of the cell. And now what else is there? Spindle fibers are formed between poles. Spindle fibers are formed. Then what else you can see here? The chromosomes reach at the center of the cell. Now what happens? The each chromosomes along with its sister chromatid, each chromosome along with its sister chromatid get attached to the spindle fibers. Each one is separately. If you look at the diagram, this one on this spindle, this one on this spindle, this on this spindle, and this on this spindle. Each chromosome separately attached on the spindle fiber. And how they attach? At the centromere. At the centromere, there is a special kind of protein found that is called kinetochore, and the kinetochore is the substance which allow them to attach. Okay, so this is how they will form the that is attachment. Okay. Okay, the spindle fiber okay, and this is attachment okay. Now the next is the chromosomes become short and thick and most clearly visible under microscope, normal school microscope, light microscope, which we are using in the school. In that also you will be able to see the chromosomes. Okay, next comes the anaphase. Now what happens in the anaphase? The spindle fibers are already formed, chromosomes are already arranged center of the cell. Now comes the most important part, that is the separation. M phase, that is mitosis or meiosis, is actually meant for the separation of the chromatids, sister chromatids. So how they will be separated here? So the spindle fiber, as I already said, has an ability of contraction. So here the spindle fibers will start contracting. As they contract, the two sister chromatids which were attached at the centromere they will separate from one another. They will break apart, and each sister chromatid will move in the opposite direction. So this red and this red are the sister chromatid. This is going into that side, so yeah. So one is going to north, the other one is going towards south. Okay. So these two blue sister chromatids, one is going in the upside and the other one is going downward, and the same the third and fourth. So each sister chromatids which were found attached at the centromere are now separated as they start moving towards opposite pole, and during their movement they will take the different shapes. So see, due to this, chromosomes separate into two sister chromatids and they start moving. As the spindle fiber contracts, they start moving away from each other, and their movement will give them a peculiar shape, either V shape or J shape or I shape, that is depending on the centromere. Suppose this is a chromosome which having its centromere at the center. So when it contracts, suppose this is the spindle fiber, and when it contracts, so it takes V shape. Just reverse it, so we'll find V shape. If the centromere is towards one side, then it will take a shape like J shape. Okay, inverted J. नीचे की तरफ जाएगा तो वैसा दिखेगा. And if centromere is towards one end, then it appear like I shape. So it is only for the sake of study. It has no importance or significance. The chromatids soon start uncoiling. So the coiling is only for a short period, that is for the metaphase, and then that they will start uncoiling. Now this is the last phase, telo. Telo means away, far. Okay, this is the last phase of the mitosis, and this phase is just opposite to the phase of prophase. Whatever changes we are seeing in prophase, now telophase are just repeating it in the reverse order. So see, we can see, and this phase, the last phase of the mitosis. In this phase, the prophase is going in the reverse order. The chromatids start reaching to the respective pole because of the contraction. Now, how they become the opposite to the prophase? You see, the chromatids start uncoiling. There we are seeing the chromatids were coiling. Here they are uncoiling. Then the nuclear membrane forms once again. There the nuclear membrane was dissolving. Here they are forming again. Nuclear membrane, nucleus also reappears. There they were dissolving, and the centrioles divide into two centrosomes. Okay, so here two centrioles they will divide and they belong to two distinct centrosomes. Okay, so cytokinesis is the next. Okay, so telophase, at the end of the telophase you will have two nuclei in one single cell. Two nuclei in one single cell, and we know that most commonly a cell have one nucleus, except in some example like in muscles, striated muscles, you may have number of nuclei. In fungi you may have number of nuclei. In paramecia you have got two nuclei, but generally each cell have got one nucleus. Large number जो है, that says that one cell has got one nucleus. Here two nuclei are formed in one cell. So now the nucleus is to be separated, and when the nucleus is separating, it is also taking a certain amount of cytoplasm with it. So that is called cytokinesis. Now the process of cytokinesis will be different in the plant cell and the animal cell. So let us talk about the cytokinesis. Cytokinesis is the process of division of cytoplasm. Okay, in this process, a certain amount of cytoplasm along with the newly formed nucleus and cell organelle are forming separation and new cells form. Okay, the process of cytoplasm is different in the plant and animal cell. Let us talk about cytokinesis in plants. So in plant cell, how it will form? Suppose this is a plant cell. Now we know that in the plant cell there is a cell wall. So suppose this is the cell wall, and at the end of the telophase, two nuclei are formed. These are the two nuclei, and the chromatin material uncoiled and all. So these are uncoiled chromatin material. Okay. Now, how that is cytokinesis begins from the center of the cell. There are certain type of cell plate formation begins. Okay, cell plate formation begins at the center, and this cell plate formation will continue from the center to nucleus, center to periphery, and ultimately it will divide the cell into two, and then cell membrane will form inside. So this is how you will find two distinct cell will form in plant. The cell plate which was there, now it will be known as the primary cell wall. Okay. Where the raw material for the cell plate formation comes, it is actually polypeptide which is coming from Golgi body. Okay. So here the cell plate formation begins from the center of the cell and moving toward the periphery. So this is called centripetal movement. Okay. So this is the and joining on the cell membrane, this will separate the cell into two different cells or two daughter cells. Okay. So this is resulting in the two new cells and all. Okay. So that is you can pause the video and you can see what is written if you have to make notes. Okay, cytokinins in animals. In animals, what happens? Animal do not have cell wall. So in animal, the process is rather simple. How? Suppose this is a cell, and these are the two nuclei, one and the two. This is its centrosome. This is its centrosome, and here the chromatin material, and these are the nucleus. Same is here, and nucleus. Now what happens? The cell membrane start moving inside. Cell membrane start moving inside from all sides. Okay, you do one thing. You take a balloon, fill air in it, not full, but little less air, and then you start twisting in it. So what you find that balloon start moving inside from the center, and as you complete the twist, the balloon will divide into two equal halves from the center, and they are attached. The same way, this movement from all the sides will be going inside and inside, and then finally a stage will come where they will join together, separating the two cells from one another. So this is how cytokinesis in animal cells take place. So you see at the end of the telophase, the cell membrane from all sides form from the center, start moving toward the center. Gradually they meet at the center, dividing the cell into two equal parts. This results the formation of two exactly similar kind of cells. So cytokinesis in animals is rather simple than the cytokinesis in plant. Now significance of mitosis. As I said, if mitosis is there, then why meiosis? And if meiosis is there, then why mitosis? What makes mitosis different from meiosis? There is some significance. So what is the significance? What is the significance here? The significance is that a mitosis is producing exactly similar cells. The two daughter cells which are formed are exactly alike. Why? Because they are separating the sister chromatids which are formed by the replication of DNA. So whatever genetic material is in cell A will be there in the cell B, which are formed from one another. So they will be exactly alike. So they will be capable of carrying out all the activities which are cell A is able to carry out by the cell B. Okay, so all these cells which are there in my body, they all come from one single cell, that is zygote. And these 300 trillion cells which are there in the body, each cell is having the same type of genetic makeup. And from those cells of genetic makeup, they are able to make the different parts. And you see the color, see the capabilities, see the retina of the eye, see the iris of the eye, see the shape of the eyes, see the thickness of the hair. All these things are exactly same in all the cells. Later on, in due course of time, they may develop mutation and they may show some variation. But generally, they are generally they are alike. So this is the significance of mitosis, that they are forming exactly similar type of cells. So they will have 100% same capabilities. Okay, so whatever my this side cell is there, that the same type of cells will be here. What is my shape of the ear there, that same type of information in the cells of this side ear will have. So there will be capable of carrying out similar type of abilities. Okay, so the daughter cells will be able to carry out all these functions with which parent cell were able to do. Uses of the mitosis. Mitosis काम क्या आता है? Mitosis काम आता है growth के. Mitosis is important for growth. Whether it is in plants or animals, you see plant how fast they grow. Animals rather they are slow growth, but growth is there. And growth means addition of new cells, and the new cells come from mitosis. Development, that is the formation of new tissues, organ systems, and body. That is all taking place by mitosis. Wherever new cells are required, that all will come from mitosis. Growth and development and repair. There are sometimes wounds are formed, sometimes some other damage is there. So damage are repaired by mitosis. Regeneration of the lost organ, like Hydra may lose its tentacle, or Planaria may lose its some part. These will be regenerated, or lizard may lose its tail. So they will be regenerated by mitosis. Reproduction. There are two types of reproduction: asexual sexual reproduction. Asexual reproduction itself is of many types. All these type of reproduction are taking place by means of mitosis. However, in meiosis, there is a separate type. Asexual reproduction, there is a separate type of cell division is required. That is meiosis. That is only for the formation of gamete. But the gametes are formed in the organ called gonads. The gonads have fixed number of cells. These cells are by mitosis, they are multiplying and maintaining their constant number of fixed number. So mitosis even take place in sexual reproduction. Okay, replacement of the dead cell. Every day millions of cells from our skin is dead and they are replaced. Then the millions of cells from our intestine is eaten away by the various enzymes and the hydrochloric acid and other things. Millions of RBC are dying per second, because we know that the RBC's lifespan is only 120 days. Before that only they die. And the WBC has got all the more less lifespan. So these cells are continually dying and these are continuously replaced by mitosis. Okay. Now, another thing which we see, which is an ill effect of mitosis, that is called cancer. What is cancer? Cancer is uncontrolled cell division. See, cell division will take place only when and where it is required. Cell division is not taking place unnecessarily. Cell division is not taking place anywhere. Cell division is always taking place in a fixed required places. If I get a cut here, then here only the cell division take place. If there is no cut, unnecessary cell division will not take place. But if due to some reason or other, cell lose this ability to differentiate that whether it is required or not required, they will continuously, ongoingly, they continue to divide, then the cell will form a bunch of cell that is called tumor. And the tumor, if goes uncontrolled, then it is called cancer. So cancer is a state of uncontrolled cell division or uncontrolled mitosis. So there is a mechanism of our body that is which is preventing the cell from dividing. That is called contact inhibition. So all the cells which are here, they are in contact with one another. So every cell is having a certain amount of pressure exerting on it all the time. When there is a cut, the pressure loses, and then the cell become active and they start dividing. And this is a controlled division. Okay, when the pressure is again maintained and the cell division will stop. But due to some reason or other, if this mechanism is lost, how it is lost? It may be lost due to some high energy radiations like ultraviolet radiations, like when a person is exposed to sunlight for a longer time, or when someone is having number of time X-rays done. See, now one more thing we must remember, it is not necessary that too much of exposure is required. Even one single exposure can lead to cancer. Okay, it is just like that. You got a stone and you got a target here, a small target. Sometimes 1,000 times you hit and you may not hit to the point. Sometimes it may be possible that the one single hit you hit and you hit the target. So it is a chance. So sometimes the UV rays, one single UV rays is reaching into that particular gene which is responsible for causing cancer, and it may lead to cancer. Or sometimes millions and millions of rays will not be able to cause cancer because it is not hitting the target. Okay, so that is the thing. Then beside this, there are certain type of chemicals, certain type of preservatives, and the drugs like cigarette smoking, nicotine, and the other type of drugs. So you know that cigarette smoking is associated with alcohol, cancer. In all cigarette packets, you find that cigarette पीने से cancer होता है. Whether you are smoking, whether you are chewing, whether you are sniffing or whatever, whichever. And it's not even necessary that we directly take it. It may be a passive smoking also. So cancer is uncontrolled cell division. Uncontrolled cell division. और this uncontrolled कैसे हो रहा है? How it become uncontrolled? It become uncontrolled due to radiations, certain type of chemicals like preservatives, VATIVES, or drugs. Sometimes some viral infection also may cause cancer. Okay, so that's all for today. We have studied about mitosis, its significance, and everything. So if you like the video, please thumbs up, and if you don't, you can always give me a comment. That this is required to be improved. Okay, and if you like it, share it with others and subscribe my channel if you are new on my channel, and if you already subscribed, that's fine. That's all. Thank you. Good day. Enjoy your day.